I want to take this opportunity to introduce you to our work on natural and synthetic glycan arrays, which can be used to examine interactions of the innate and adaptive immune systems with zwitterionic glycans. For those who are perhaps not so familiar, I just want to say there are many different types of glycans or glycoconjugates in animals, ranging from proteoglycans through to glycolipids, glycolipid anchors, and glycoproteins with the glycans linked either through asparagine, that means N-glycans, or through serine or threonine, means O-glycans. In animals, there are a wide range of building blocks for these glycans, and they are represented by color and shape codes as internationally agreed, as shown in the box on the left-hand side, and this is what I'll use throughout the rest of the talk. The focus of our work is on N-glycans in lower animals. So, for instance, in mollusks, we have found sulfated blood group A, glucuronylated fucose, monomethyl aminoethyl phosphonate, in nematodes, phosphorylcholine, fucosylated antennae, or even in one species, glucuronylated antennae, and in arthropods, that means in this case we've examined insects, glycans with phosphorylcholine, phosphoethanolamine, and glucuronic acid on them. Now, aminoethyl phosphonate, phosphoethanolamine, and phosphorylcholine are zwitterionic modifications, which means they have a positive and a negative charged character. And our studies do show that many lower eukaryotes do modify their glycans with these groups. PE and PC are more familiar perhaps as the head groups of phosphatidylcholine or phosphatidylethanolamine, or as the modifications of bacterial lipopolysaccharides. And so it's not perhaps surprising that as they are foreign to humans in the context of being part of glycoconjugates, that PE and PC are recognized by immune system, for instance by pentraxins. That means proteins such as human serum amyloid P or C-reactive protein. C-reactive protein, or CRP, perhaps you're familiar with as one of the values when you have a blood test done, is a marker of inflammation and can activate, for instance, the complement pathway. When we looked at the honeybee in terms of the glycome, we first looked at royal jelly. Now, royal jelly has almost a mystical status, as it's been used as a beauty or anti-aging product, but is, for the bee, important as a sole food for the queen bees. Now we analyze the N-glycans by offline Molotov MS. Striking was the presence of phosphoethanolamine. This modification, which is 123 Daltons, is sensitive to hydrofluoric acid, and this sensitivity can be shown by shifts in HPLC retention time or in the MSMS fragmentation pattern. Some glycans contained phosphoethanolamine and glucuronic acid at the same time, and this gives rise to a fragment of 866 mass units. Now, so much for the structure. What about potential function? We took the natural glycans from raw jelly and modified them with a linker called AEAB, which on one hand can bind to the glycan, and the other hand can be printed onto special glass slides. First of all, we used standard plant lectins, concanavalin A and peanut agglutinin, and found nice binding, particularly to the neutral glycans. And so you can consider a structure as shown here, that the mannose residues could bind the con A, and the GalNAc modification, the PNA. Now, what about something which might bind zwitteronins? So here we took SAP, which is known to bind phosphoethanolamine, and saw there's a nice binding to the anionic fraction. And this would correlate with glycans such as that one shown, with the PE residue and the glucuronic acid. Next organism I'm going to talk about is Dirofilaria immitis, or the dog heartworm, which is transmitted via mosquitoes. And the geographic range of Dirofilaria is increasing due to climate change. Why is it called a heartworm? Well, the adult stages accumulate in the dog heart, and these worms have to be removed surgically. And this was the source of our biological material. We found a wide range of interesting glycan structures with fucose residues, phosphorylcholine residues, and glucuronic acid residues too. In this case, the zwitterionic modification is phosphorylcholine, and this can also be removed by hydrofluoric acid, leading to a reduction in mass of 165 Daltons. But this treatment also removes particular fucose residues, as also diagrammatically shown here. Now we took pools of glycans, either not treated with hydrofluoric acid or treated with hydrofluoric acid, labeled them as AAB, and then put them on the glass slides, and found very nice binding to C-reactive protein. Now it's regardless of whether we had glycan on the slide, then C-reactive protein, then anti-C-reactive protein, or we had glycan on the slide, C-reactive protein, C1q, which is the first component of the complement cascade, followed by anti-C1q. If we fractionated the glycans with regard to size, we could find that the largest glycans are those which most recognized, the CRP. Then we had a look at the sera of dogs, in particular looking at what IgM and IgG was present. So somehow the reverse pattern as compared with CRP. The natural glycans did have a signal, but the HF-treated glycans have a higher signal to the IgM as well as the IgG. And this seemed to be regardless of whether we had the one non-infected dog or the two infected dogs. So it could be that the natural glycans help the parasite to hide from the adaptive immune system. Now, as these glycans can be quite highly branched, it might be that the distances that you can calculate between the different antennae could correspond to the distances between subunits of CRP in the pentraxin pentamer, or between the FC arms of antibodies. Now, with natural glycans, there is a little bit of a problem in that you have low amounts and high heterogeneity. So we also employed synthetic compounds. Here either mannose or the disaccharide glucan mannose, with or without PC or PE on a special linker. There were also comparisons with lacdinac and chitobiose. That means galnac or glucan, as these are also components of nematode glycans, and a couple of other controls. Now, the con A bound very well to the man and the glucan man, but binding was abolished if you had either the PE present or the PC on the mannose compound, or reduced if you had the disaccharide modified with the PC. In contrast, C-reactive protein, or the well-known anti-PC antibody TEPEC15, or the anti-PC antibody 6G3, which is derived from Trichinella-infected rats, bound only to the PC-containing compounds, regardless of whether it was the mono or the disaccharide. And then we also looked at IgM and IgG in the sera of animals. And here we see actually a complicated pattern that either we have infected Ascaris, or pigs infected with Ascaris, or dogs infected with Dirofilaria, and that there are variations even between individuals in terms of whether you have anti-PC binding or not. So, I'm hoping I convinced you that there's a wide variability in N-glycan structures between invertebrates, that some of these structures contain zwitterionic modifications, that these zwitterionic modifications are recognized by pentraxins and also by antibodies. And finally, I'd like to thank various members of the team for their work on this project, as well as our collaborators, funding agencies, and you for your attention.